All right, well, you've seen the theme emerging that from both Doris's and Christine's talk of the visual system constructing an internal representation of an external stimulus. And I've always loved Christina's work in this area, dating back to the 2001 paper looking at this at the perceptual level and demonstrating more recently that she can causally perturb this. I really feel like these two talks represent a type of neuroscience where we're really getting at that process of constructive perception. Right now I'm introducing Arash Afraz, who is going to take a very different perspective. He does think about the visual system and perceptual system as being related to these sorts of processes, but he exploits artificial intelligence in a very different way as the enemy. And I won't steal his thunder, but you'll see this emerging in his talk. Welcome, Arash, who is a professor at the National Institute of Mental Health.
All right. Well, I hate to be the only thing standing between you and your lunch, and I'll do my best to distract you from imagining me as a roasted pig. All right, so let's get into it. I couldn't have asked for any better setup than Christine's talk for what I want to say. So as she mentioned, there is perception and there is neural activity. And if we want to think about what makes perception and how perception is generated by the brain, we need to bridge this theoretical gap between perception, which is something that is measured in psychophysical units, it's a form of behavioral report, and neural activity, which is measured in units of spikes per second. And to do this, we know how to measure neural activity, but how do we measure perception is a challenge. This is a neuron that is recorded by Bob Desimon in actually 40 years ago exactly. And we know about these sort of receptive field properties of neurons in high-level visual cortex. This neuron responds most to this monkey. It responds pretty much to Bob's face. This is how Bob Desimon looked back then. And here there is some sort of response to his famous toilet brush. And so we kind of don't know what is exactly driving this neuron, and we have been studying the hell out of that. One thing that we have not been studying is a concept. I actually termed it after a 1988 paper by Terry. I hope he doesn't mind borrowing this concept. I call it the projective field of a neuron. How does that neuron project into behavior and perception apart from what it receives from the external world? So we just don't know, simple as that if we stimulate that neuron, would the monkey perceive the best stimulus that was in the set? Or maybe the monkey perceives Bob, because that's more biologically important for the monkey, is who gives him food. Or maybe a mishmash of all those features. Or maybe the monkey imagines his late aunt, who hasn't happened to be in the stimulus set. Or maybe nothing. Maybe the code is so distributed that the monkey would not perceive anything, and we cannot really change the perception by a local perturbation. So the answer to this problem is quite critical, as building visual prosthetics actually depend on knowing the answer to this question. Visual hallucinations in mental disorders, I'm coming from NIMH, and we care about that. And perhaps in a more abstract concept, building a mechanistic theory of vision requires solving that problem, how to map neural activity into perceptual events. So the way we have been measuring this is by using psychometric functions. And this starts, as Christine mentioned, by early works by Newsom's group, and many other notable works that I have dropped actually here, and that includes my earlier work on face detection, that we show this sort of shift in the psychometric curves. But when we work with objects, actually, even in 2006, I had this problem that, okay, my monkey seems to be perceiving a face, but what face is that when we stimulate the face neurons? Is it a face? Is it a monkey face? Is it a human face? Is it a long face? Is it a narrow face? Is it a happy face? Whatever. There are many possibilities. And what determines that? So the problem is that dimensionality of perception is really high, especially when we get into complex object perception in high-level visual, in like IT cortex. And what happens is that we kind of make this assumption that this perception change, this perturbation in this high-dimensional space happens, which is represented here by the red arrow. But us experimentalists cannot capture that. So we kind of take a projection of that vector into something we can measure, let's say on a context of a face, non-face task, and we measure the projection. But we miss the big picture, we miss the original. And what I want to try to convince you is that it is possible to actually measure the red vector. And as I am talking about this, that we want to understand if we perturb the brain, what exact perturbation happens in perception, and can we take a picture of that, you should be starting thinking that he is showing signs of middle age onset schizophrenia or something like that, because perception is subjective. How can we interrogate that scientifically? How can we take a picture from what the monkey perceives? So before that, let me talk a little about the toys that we are using here to interrogate perception. We have been using, as Christine mentioned, she actually saved a lot of my time to pitch why it's better to do it optogenetically. So we have switched to this sort of optical array that we developed a while back. And this is an array that you can implant on the cortex following injections, virus injections in the cortex. We have published the protocols in this paper if anyone is interested. We open up the cortex. This is a base of brain surgery. We inject virus on an industrial scale with a multi-needle injectrode. And we kind of inject a lot of virus in the cortex, close the monkey down, come back in a month, open up the dura again, and confirm there is fluorescence, there is expression of the virus. And we use that fluorescence in the OR to align our optical array and implant the array on the cortex. Now we have a plug-and-play monkey, and we can collect actually high throughput behavioral data with optogenetic perturbation of the cortex. So first of all, our question was, would the monkey detect cortical stimulation? Forget about asking the monkey what he perceives. Can he see local perturbation in IT cortex, local optogenetic perturbation in IT cortex? So here what you see is the training profile of one of our monkeys who is supposed to detect stimulation of a cortex, basically turning on one of the LEDs on the LED array. And this is the y-axis, is the proportion of stimulation report, and x-axis is just time. And these two lines show stimulated versus non-stimulated trials. As you can see, the monkeys very easily and very quickly learn this task, and they can separate between stimulated trials and non-stimulated trials, to the point that we were worried maybe the monkey sees the light from inside the skull. It goes in the back of the retina and hits the monkey's retina, and that's how the monkey detects it. So we actually put another array in the opposite side, opposite hemispheres IT without injecting any virus, and did the same protocol, and that basically makes these catch trials. The monkey actually categorizes them as not stimulated. So this is an example of the task that we have been using. We call it the CPD task, cortical perturbation detection task. So what we do is that we ask the monkey to fixate on the screen, then an image, could be anything, is shown on the screen for 1 second. In the middle of that 1 second, for 200 milliseconds, we just roll the dice randomly, either do nothing or turn the light on IT cortex for 200 milliseconds. Then the monkey is supposed to make a saccade to one of the targets to report if it was stimulated or not stimulated. And we reward the monkey based on that rule. So the first thing that we noticed was that, basically the first question that we asked was that, does something visual happen to the monkey when this happens? And our proxy to measure that first was to choose different images on the background. These images are not informative of the condition, task condition. If I am putting a rat there, the rat is presented in both stimulated and non-stimulated trials. So the object on the screen is a mere wallpaper. It doesn't have anything, it doesn't have any information for the monkey. But we thought that maybe the monkey perceives some change on top of this image when we stimulate the brain. So here is the earliest result that we found. Here on the y-axis, you see the monkey's performance in the CPD task, the monkey's ability to tell the difference between stimulated trials and non-stimulated trials. On the x-axis, you see the different images that we have used. And to our surprise, there was crazy variation. For some images, it was very easy for the monkey to detect cortical stimulation. For some images, it was very difficult to do so. Why? First, we thought that maybe, let's say, imagine the monkey perceives a red star. Well, imagine it's like a lefty monkey. Actually, one of my monkeys' name was Castro. So imagine Castro sees a red star when you stimulate the brain. You can imagine if your object happened to be red, it would be harder for the monkey to detect that on that background. And if the object is yellow, let's say, it's easier for the red to pop out. So our question was that, is it coming from some sort of a crowding effect, some sort of an image effect on the screen, or is it really coming from the mind? So what we did was this experiment. We took a bunch of images and faded them to gray. So now the question, and we performed the same task on all these gray levels, on these different contrast levels. So now the question was, would the monkey perceive the cortical stimulation better for the high contrast stimulus or for no contrast stimulus, for the blank slate? Initially, I thought that if the monkey has a constant perception that is coming from the brain and is not dependent on the image, doesn't matter, we should get a flat line, right? And we call that the basic hallucination hypothesis, that the monkey hallucinates something additive, something is added to monkey's perception, or we would see a line that is going down actually because of the interactions, crowding effect with the image. The alternative hypothesis was some sort of distortion, that maybe the monkey perceives a distortion which is relevant to the object that is presented. That would expect actually a crescendo plot. And that's actually what we observed, which was quite surprising for us at the beginning, because I thought first that if you give the monkey a blank slate, it would be the purest form for the monkey, easiest for the monkey to detect cortical stimulation, that red star or whatever it is, right? But it turns out not to be the case. When there is no stimulus, it is hard for the monkey to detect the stimulation. And we dug into human stimulation literature, and we found this paper from Morphy and Mansell that actually shows that even in humans, if you blindfold them and stimulate IT cortex, they see nothing. And that was quite amazing in terms of basically where this research took us. We didn't really believe this, so we thought we should do it again with another technique. So this is works of Rosa Lafersuza, my wonderful postdoc. The other work was by Reza Azadi, my other postdoc at the time. So this one, what Rosa did was now to she didn't change the contrast. She just varied the size, made the image less and less visible by just shrinking it on the screen. The idea was that if the distortions are limited to the object itself or somehow stuck to the object, it should be harder for the monkey to see them. And again, we found that the bigger the stimulus on the screen, the easier for the monkey to detect cortical stimulation, which again suggests something is happening to the phenomenologically at least, something is happening to the object that the monkey is perceiving. So this actually triggers the question that maybe we cannot drive the neurons equally, even though we are using the same power, the same optical power to kick the neuron in both conditions, in all of these conditions. Sometimes maybe we can't move the neuron as much. Maybe you can imagine, because these neurons are pushing and pulling each other. So they are living on a manifold, and maybe that actually creates these impermissible spaces. Let's say you get two neurons that are inhibiting each other, right? Getting them both active require either breaking the circuit, or basically you won't be able to go there to the point that they are both active. So maybe activity of neuron 2 actually determines perturbability of neuron 1. Maybe you need to get the neuron active to actually get your stimulation to work. Maybe when you don't present anything to the monkey, the brain stops neurons from, anchors them down, because the rest of your brain is telling you there's nothing on the screen. So far, I've told you that brain stimulation in IT cortex create percepts that are probably have a distortive nature. They are dependent on the contents of concurrent perception. But I haven't told you how to measure the red vector, and that was the goal. So now let's dive into that. This is a set of works that we we named them perceptography. It's a technique, it's a concept that works as following. We get the monkey to fixate on the fixation point. We present an image, and we get the monkey to do the same task. If we stimulated your brain, you got to look at that dot. If we did not stimulate your brain, you got to look at the other dot. But now, in the middle of the experiment, we cheat on the monkey. Actually, we change the stimulus. We perturb the image. The idea here is that if just give me the benefit of the doubt, I know that's impossible, that's very unlikely, but if we happen to warp the image in the same direction as the monkey perceives when we actually stimulate him, we might get the monkey to make a mistake of false alarming, of reporting a non-stimulated trial as stimulated. And that's actually what we hook into, and we get into the perception. The logic is as follows. Imagine the monkey perceives Moby Dick when you stimulate his brain. Right now, if we show Moby Dick here, right, exactly as the monkey perceives, then the monkey cannot tell the difference between the stimulated trials and the trials in which he was presented with Moby Dick. So now the question turns into a search problem. Instead of that philosophically closed problem, now it's a search issue. How can we find Moby Dick in this ocean of infinite image perturbations? So the way we do it is that two AI systems control the experiment under the hood and function as adversarials to the monkey. The first system is Da Vinci. We call it Da Vinci. It's a kind of GAN. It's a kind of generative adversarial neural network. What Da Vinci does is that it takes that base image and mutates it in random fashion. Thousands of mutations. It just spits out thousands of mutations of that base image. And now the monkey responds. Now there is another guy, another deep neural network. We named it Ahab after Moby Dick's Captain Ahab, because he was after Moby Dick. So we thought Ahab should actually count the number of false alarms by the monkey. And now Ahab's loss function is the false alarms of the monkey. Ahab's job is to talk to Da Vinci and tell Da Vinci, you know what, you put a white spot there in one of your mutations. That turned out to make the monkey false alarm 1% more. Maybe there is something in there, that part of ocean. Can you get me more of those images? So this loop between the monkey, Ahab, and Da Vinci continues for thousands of trials, during which the monkey trains the machine to fool him, basically, because the machine is after monkey's false alarms, and the monkey doesn't get reward for false alarms, right? So he has all the incentive to sharpen his decision and tell the difference between stimulated and non-stimulated trials. So now, let me show you an anecdote of the first day we switched to this paradigm, because it's a cute little anecdote. These monkeys are trained first on the CPD task, as I mentioned. There's a stable, solid image on the screen, and then you zap the monkey in the middle, right? The first day that we start perturbing the image at the time of the brain stimulation, this happens. This is the false alarm rate of the monkey, and this is the trial numbers. Up to this point, we just use the static images, and false alarm rate is pretty low. The monkeys are pretty good at doing this task. All of a sudden, as soon as we start perturbing the actual image, the monkey's false alarm rate skyrockets. It clearly shows that first, the monkey sees something on the screen when you perturb the cortex. Second, it shows that the monkey's internal rule for doing the task is that if the image changes, I got to look at that dot, and this guy rewards me. If the image remains unchanged, I got to look at the other dot, and this guy rewards me. Now that we introduce changes in all images, the monkey has to sharpen that criterion again and say only Moby Dick counts, not all images count, right? They get it. Within a few hundred trials, the false alarm rate drops down back to what it was now in the face of those perturbations. Now the monkey is looking for only the Moby Dick, what is coming from the brain stimulation. And now Ahab can get into work by basically training itself against the monkey. So we were worried that if this paradigm really works, we are untraining the monkey, right? Because the monkey is doing the right thing and we are not rewarding him. So he'll just drop doing it altogether. To keep the monkey hooked into the task, we diluted Ahab images with lots of Da Vinci images, lots of non-optimized images. To get the monkey in there, that's against us in terms of power, but it helps us sneak those Ahab images in the middle of those other images. And what we see is that even though these images are interleaved, for Ahab optimized images, the false alarm rate starts to go higher and higher and higher. So now, this research was done in the peak of the pandemic. So we were looking lots of virus evolution plots and dendrograms. So in the spirit of the pandemic, we plotted a lot of our plots in this fashion. You can think about this process as an evolution. These are the different mutations of the base image, of the seed image, the original, and these are different iterations of the image evolution. Basically, any image that has created a false alarm has the chance to reproduce and go to the next generation, has some children, and then we continue applying that rule until these images evolve that create really high rates of false alarms. Our threshold for that is 60%. So remember, baseline false alarm rate is like 3%. So if it crosses the 60% false alarm rate, we call it a perceptogram. But we have seen images that create 100% false alarm. This is an example of an image here. This is the base image. This is a weird GAN-generated image. It's a bunch of towels with a finger on top of the towels. And these are the mutations that Da Vinci has created. This guy happened to create more false alarms. The thickness of this line here shows the false alarm rate. And then this sort of evolution has gone and gone and gone until we got into this perceptogram, which induces more than 80% false alarm in the monkey. In that sense, the monkey has a really hard time telling the difference between the state of looking at that image, perceptogram, and the state of being stimulated in IT. We are like, is it really true? Let's do it again. Let's throw out all of the data and start another evolution with the same point, the same seed image, and we converge on almost exact same perceptogram. So what we observe here is not a local minimum, it's a global minimum, and you can sort of get back there again. Let's take a look at a few examples of these perceptograms. What kind of images are they? Some of them don't make any sense. Some of them make some sense. And I'll sort of get back about how to organize them a little later. But this is a wild example. That's the cat. And that's the perceptogram of the cat when you stimulate a given point in IT cortex of a macaque monkey. You see these noise patterns, this sort of mushroom-like experience is happening here. You see these bright noise patterns on top of the cat's face, and these are very specific patterns. If you move this pink streak here from here to there, the monkey is going to notice it because the monkey is looking for any opportunity to do the task right and get reward. So this is noise, but it's a very specific, unique noise that happens over and over with the stimulation of that point in the cortex given this image on the retina. This is an example that makes sense. This is an example that is more on the natural manifold of image changes. This is a monkey seed image, and when you stimulate a point in the cortex, in IT cortex, that monkey seems to appear as sort of another monkey, maybe same monkey with the head 5 degrees rotated and the hair growing long, and I guess that's platinum blonde. So that's how the monkey perceives this, which is sort of a more natural change, which makes sense. Before talking more about this, I want to put this in the context of, because we are talking about generative versus non-generative brain, I want to put it in the context of labeled lines. The way I thought this system works was based on labeled line hypothesis. We are familiar with that sort of thinking. It's a concept that comes from early sensory processing, like you get a taste bud that responds to sweet taste. Whenever that guy is active, it means there is sugar around, or some sweet taste is around there. So that's a labeled line for coding that. The somatosensory homunculus is another one. Like you stimulate here in the brain, you feel a tingle on your thumb. And that's sort of the idea of a labeled line. Following that logic, I thought that IT cortex should be pretty much the same. You get these columns of different feature selective neurons, and now if you get one of them and stimulate one of them, you should be adding the best stimulus of those images actually to the screen. I would say actually that labeled line hypothesis has created a dogma that has forced us into this direction, this rabbit hole of asking what is the best stimulus, what is the preferred stimulus of an IT neuron. And I am beginning to think maybe, forgive me for my blasphemy, but maybe that's an epiphenomenon. To what is actually happening in the brain. We can talk more about that. But what we find is far from this. This predicts that if you stimulate a given column in IT cortex, you will get the same features added to the image no matter what the background, no matter what the seed image. We have a really hard time finding these features because remember, perceptograms are actually quantitative measures. These are parameterized images, so we can compare them. And we find a hard time to find same features on top of the base, so different seed images, different background images. Although there are some anecdotes. So I can show you this anecdote here, which is an interesting way to kind of conclude this talk. This is a case. This was a point in one of my monkeys, IT cortex. By the way, this work is done by Ilya Shahbazi, a great postdoc of my group. It's a dog image, and you know, this can warp into anything. This can warp into a white whale. Da Vinci is not limited. Da Vinci can create anything. But what happens is that you get almost exact same image with the tongue coming out. That was interesting because that's sort of an unmanifold change. And first, again, we didn't believe this. We were like, okay, let's make this dog 3 times larger and see if we see the same. Would the tongue appear at the same position on the screen or where the muzzle is, right? We found the tongue coming out again on the large image. And now we are like, okay, what happens if we show that image to the monkey? It's very interesting. We tried a flower, a daisy flower. Nothing happened. The monkey had a hard time detecting a stimulation, actually, when looking at that flower. And the perceptogram was the same flower slightly changing. But now we show some skin here. And we get that image. Do you see a tongue? This part, many people describe it as a tongue. So now imagine what is happening here. Yes, the same feature is added to the image in this case. Excellent, yay. But what is happening here is that the brain should come up with a plausible explanation about the world. In this case, a tongue. In the case of the dog, it's pretty easy, actually. The dog and the tongue, it's just you just add the tongue. But if you have that, and the flower cannot have a tongue, so that neuron is anchored down. You can't really drive it. But with the finger and the skin, maybe some other neurons, body-related neurons are firing, and now the tongue is possible, but now you have to build the whole face to explain the tongue, because tongues don't hang from fingers. So that's sort of an anecdote, at least, to show there is some generative processing which cares about plausibility of stimuli in the external world. This is my last plot. This is trying to make sense of why some of them make sense, why some of them don't make sense. And we found that the ones that make sense, these are seed images that we used on posterior part of the array, middle part of the array, and anterior part of the array. So we are moving on the AP axis, we are moving forward in the cortex. And what happens is that perceptograms that are achieved from posterior part of the cortex are those specific noises. They are noisy, like this rabbit's face is totally destroyed here, or this doll's face is kind of noisy, just doesn't make common sense. But as you go anterior, you get changes. They are different from the base images in meaningful ways. Like this one, this ear is more visible. This one is more crisp than that. But these are on-manifold changes as you go anterior in the cortex. So we are hoping to add neural recordings to what we are doing, and finally basically building the bridge by predicting the causal contribution of neurons to perception by measuring both neural responses and the perceptual distortions that come. And let me finish here and thank you and thank my wonderful group.
So that was absolutely beautiful, Arash. I am curious to know, in your view of how this is happening, what you predict about the firing rate. So if you stimulate a particular neuronal population and you have deduced through your perceptogram what that stimulation is doing, would you predict that if you record with the perceptogram itself, you would see an elevated firing rate relative to another stimulus that you started with, for example?
That's a fascinating question. And that's a question that the only reason we haven't done that yet is technological limitation, because doing both stimulation and recording in a chronic fashion is a headache in large brains. And we are going to do that soon, hopefully. My null hypothesis is that yes, that's the case. But then again, I am interested in kind of, okay, now let's use the perceptogram as the seed image, and let's say that it probably drives the neuron. It should. That's my null hypothesis. But now, if you show that as the seed image and stimulate the neuron again, would you be able to move it further in a given direction or not?
Yeah.
Right? And we don't have the answer to that.
Beautiful. Question right here.
Yeah. My question is, so to produce those perceptograms, you use different seed images because a blank screen just wouldn't work. I was wondering if you can start with some kind of a universal seed image, like just some spatial gradings or something, or an average image, so that all the perceptograms can be based on the same seed image?
Absolutely, we can do that. You know, the engine that we are using, this is actually, I use this opportunity to show this plot. We can start from any image, like this is an image, and now our loss function is similarity to this target image and go from there to there. So we can warp. Our image generation engine can create any image. So we can go from anywhere to anywhere. However, I think using different seed images is an opportunity, not a problem. We actually want to see if you set up the state of the brain on a given image, right? What does a given image mean? A given image means a unique pattern of activity in IT, right? You change that image, you get a different unique pattern of activity in IT. And the idea is that when you are in pattern A, how much can you move this neuron? When you are in pattern B, how much can you move this neuron? And if you think about perception as something passive and feed forward, you should be able to move the neuron no matter what. But if you think that something is being generated to explain the incoming stimulus, under some conditions, that guy should not fire. You know, if this is a daisy flower, you tongue neuron better shut up. You cannot be fired. This is not in my model. There is no flower with a tongue, right? So it makes it harder to move that neuron. So in that sense, it's an advantage, actually, to interrogate what I like to call it, you know, I like to think that vision is not taking the information from the external world and representing it after going through a bunch of filters. I think vision is making a model and consulting with the incoming signal to constrain that model.
So for the seed image, you know, to generate those perceptograms, what would you say is a good set of seed images? Like maybe 50, 100? How many semantic categories that they should encompass?
Oh, you mean how do you select your seed images?
Exactly.
We go random. We didn't want to introduce any biases in this study, so we went totally random. However, now that we kind of have this proof of concept, it is time to do that sort of studies. Like we have just launched a study in the middle face patch, because now we know these neurons are face-preferring neurons, and faces are great stimuli, as Doris mentioned, in terms of being able to parameterize them, right? So you can ask more parametric questions from there, and other categories as well. We started with faces because they are easier to deal with, and they are probably a better model for the rest of the system.
Thank you.
Sure.
In the back, Jenny.
Hi, thank you for that wonderful talk. I was wondering if you looked at the relationship to the temporal timing of when you stimulate. So if you stimulate at a different time, do you get different effects?
Oh my God. You're pushing me into revealing something that I didn't want to reveal because it's too crazy, and I have only one monkey. So forget this after I just said that. But we've done experiments on time, actually, because what you're asking is a very delicate and important question, right? If we go back, I've actually managed to hide it, but you cut it. So here, your question is, when do you stimulate and how do you overlap that with perturbation on the screen? Do you need to adjust for delay, or do you need, what is the timing of perception? If they don't match on top of each other, it just doesn't make sense, right? So what we did here is that we went low resolution. We are like, okay, let's do 200 milliseconds. These times will overlap, and the monkey won't notice that little difference between them. So because now we want to just have a proof of concept. But now with the proof of concept, now we have gone with shorter stimulation trains and varying them in time, right? And what we are finding is actually mind-blowing. We first did this little experiment of adding a masker to the image, right? Just measuring detection performance, and we assumed that at the time that the monkey perceives that warping of a given image, if we introduce a crazy moving masker pattern on the screen, it would be hard for the monkey. So now we move the masker on time to detect when does the monkey perceive cortical stimulation, right? Now I've showed you what is it that the monkey perceive, but when is it that the monkey perceive? And the result is crazy in the sense that the monkey perceives cortical stimulation. Again, forget this as soon as I just said that, it's just for fun, but 200 milliseconds before stimulation. Before stimulation. And that is quite impressive because it shows, you know, remember these neurons are sitting behind a delay line. Something happens in the world, right? Well, we measure the delay in objective terms, in like with a rolling t-test, and we are like, okay, neural activity is above baseline in 70 milliseconds, 80 milliseconds after the image onset on the retina. But what does it mean for the brain? There is some processing time too, right? Until these neurons firing means something for the brain. Even, I would say, stuff that Doris mentioned, like those little feedback loops should go a few rounds until this neuron consolidates, right? That number turns out to be 200 milliseconds for IT cortex. And when you go from sky and stimulate that neuron, the monkey doesn't know that. The monkey makes the assumption that something has changed in the world and I have to explain this, and that something gotta be before the actual physical time of a stimulation. So if we use a really short current and if we actually perturb the image 200 milliseconds before the brain stimulation, we get the best perceptograms in terms of their effectiveness. Great question. Thank you.
Ralph?
Okay, amazing work. I just have a quick question on...
Go ahead and trash it.
No. I think it makes total sense. I mean, it's a pity only for one monkey, but I'm not going to forget what you just said. But my question relates to what you said about, you know, line label codes and how we think about what neurons do, and thinking about, you know, the most excitatory stimulus or what it represents or something, and you're suggesting that that might not be the right way to think about it without elaborating. So I just want to give you the chance to elaborate a little bit.
Oh, yeah, thank you. I actually don't want the chance to elaborate because I don't know really where that goes. The idea is that some early tuning of the neuron to the stimuli, to the physical stimuli out there, might not necessarily be what that neuron means, the activity of that neuron means. What I imagine is something like a, you know, how to say it. I was actually very happy when in the Nobel Prize I saw that Hopfield is also there. besides Jeff Hinton, because I think we are dealing with a Hopfield-like structure here more than a deep neural network structure. These neurons are pushing and pulling each other in the cortex. Even if you imagine feedback from higher level areas, neurons, as John actually has showed, neurons live on a manifold even within IT cortex. So the activity of a single neuron probably doesn't mean much. There is an ensemble of neurons that are coming up and going down, like when you get that face with the tongue pushed out. You probably have the face neurons also active, right? Because they are related to each other. So the way I think about it, I'm not sure, but I guess what we should be looking for is that maybe we should be recording from face neurons when we are perturbing this other guy. And I guess that when you stimulate this other neuron, you should get some activity in the face neurons too, to find evidence that a whole pattern on IT cortex is being regenerated. I guess, but that's just a speculation.
All right, before we thank our speaker, because as soon as we do, you'll all run out of the room, I just wanted to say two things. One of them is, graduate students and postdocs, you may see one of the speakers that you just heard or one you will be hearing wandering around out in the lunch area, and I would encourage you to sit down with them and talk to them. So take that opportunity. We will be breaking now for lunch out in the auditorium here, in the foyer, and we will begin again sharply at 2:10, so please around 2:05 start coming back into your seats. Thank you very much, and thank you for a